Thank you. So I'd like to share with you a alternate approach to gene therapy, which does not involve any viral vectors, which does not attempt to replace or correct genes, and which is not limited to the treatment of inherited retinal diseases. Eyevensys is a private biotechnology company based in France. When we are developing non-viral gene therapies as a sustained drug delivery platform to come up with new treatments for a variety of eye diseases. Eyevensys is actually a continuation of the work that has been done by our founder, Francine Bercohen, who is a vitreoretinal specialist who has worked on trying to come up with better treatments for eye diseases. So our current pipeline for Eyevensys includes products which result in the sustained delivery of known therapeutic proteins to the eye. We have OIS 606, which results in the intraocular expression of an anti-TNF protein, which we have in clinical. We have OIS 611, which results in the production of a neuroprotective protein that we will intend to develop for retinitis pigmentosa, but we also believe could be an effective treatment for dry AMD and possibly glaucoma. And then we have OIS 609, which is an anti-VEGF, which is in very early stages of clinical development, which we will develop for the usual anti-VEGF indications. So why do we need a better sustained delivery drug platform like what we are developing with Eyevensys? Well, because there are limitations to all the ways of delivering drugs to the back of the eye with current commercialized formulations. First, we know that proteins delivered by intravitreal injections don't last in the eye, and therefore you need to repeat the intravitreal injections frequently, and this is causing problems for patients as well as for physicians. With viral vector gene therapies for the treatment of inherited retinal diseases, you have to have an invasive surgery, and you actually potentially can cause damage to the retina itself, and you only treat a small area of the retina, despite the fact that the whole retina really has the genetic defect. We have implants that have been used that get products secreted in the eye all the way out to over a few years, but most of these implants actually involve corticosteroids, which we know over time will result in adverse events to the eye. And then most ophthalmologists don't like the idea of systemic medications because they have to deal with systemic side effects, and quite frankly, the efficacy of these treatments really rely on the patients taking their medications. So the Eyevensys technology actually addresses a lot of these issues. So by having the eye produce its own medication, you avoid the need for repeated intravitreal injections, and unlike viral vector approaches, you don't end up having the protein being produced in the eye for years, possibly after it's not needed. The procedure is actually minimally invasive, and so you essentially will reduce the risk of retinal injury as a result of the treatment, and there will be very limited systemic effects and very limited ocular side effects. And because we're not dealing with viral vectors, the cost of producing the plasmids that we'll be using is going to be very low in cost, and there's no problem with repeating the administration if necessary. So what exactly is the Eyevensys technology, and how does it work? Well, we are actually a drug delivery platform that turns the eye into a biofactory for secreting its own medication. And essentially, this is a combination product consisting of a biologic as well as medical devices. So we start out with plasmids on which we encode therapeutic proteins, and we can encode pretty much any therapeutic protein that can be secreted by the body. These plasmids are then injected into the ciliary muscle, which is the site of our biofactory, by using our electrotransfection system, which was proprietary to Eyevensys. So we've developed these ourselves, and they consist of two components, an ocular device as well as an electrical pulse generator, which together are used to create an electric field, which then helps the plasmid, which is injected into the ciliary muscle, make its way to the nucleus, where the protein encoded on the plasmid is then expressed and secreted into the vitreous as well as the choroid. And that is the way that the protein can then affect its extracellular target at the retina. We validated the Eyevensys technology in animal models where we've shown you can safely administer plasmids to the eye. You can repeat the electrotransfection on the eye without causing any damage to the ocular structures, and we can produce a wide variety of proteins in the eye durably for a period of up to 9 months in animal models. But what's more important than just being able to produce the protein is to be able to know that the protein actually has an impact on the disease that you intend to treat. And so we've been able to show with our anti-TNF proteins that we've been able to reduce the severity of uveitis models. With our anti-VEGF proteins, we've been able to, in laser models of CNV, reduce the leakage. And then in our neuroprotective proteins, we've been able to actually show that in a variety of neuroprotective models, you can actually preserve the structure and the function of the retina. So we are currently in clinic with our lead product, which was EYS-606. And in animal models with EYS-606, we've shown that you can reduce the severity of EAU, which is a very close model to posterior segment uveitis. And you can also, in another model of uveitis, you've been able to show that the effect of EYS-606 is similar to that of dexamethasone. So with this background, we actually started a phase 1-2 clinical study showing the effect of EYS-606, and I'd like to share with you the results from the study. We had no safety concerns. The treatment was well tolerated, although we saw adverse events that were mostly related to the treatment procedure. We actually saw that the profile of the administered treatment was very similar to other drugs that are administered intravitreally or intraocally. We did see some efficacy findings, including improvement in visual acuity in one patient in the first cohort that was timed at the peak of the onset of the drug, and then we also saw a reduction in macular edema, improvement in visual acuity. And here you see the baseline OCTs in 2 patients that were treated in the highest dose cohort, which showed very thick macular edema, but at 6 months there was a reduction in macular edema in both of these patients with improvement in visual acuity in one patient. And after the study, a patient that didn't have improvement in visual acuity was actually seen again 2 months after, and this patient showed another interval improvement in the OCT, as well as now an improvement in visual acuity of 14 letters. And so the evolution of the OCT changes in these patients actually supported the fact that the protein is being expressed in the eye. So our next product that we have in preclinical phase is EYS-611 for the treatment of retinitis pigmentosa. And we've been able to show that with EYS-611, you can preserve the function of the retina. And even compared to other treatments that are in development for retinitis pigmentosa, we preserve better the function of the retina, as well as we have been able to show preservation of the retinal function. And different from EYS-606, we've actually been able to show with the same concentration of plasmid, you can produce higher concentrations of protein in the eye and sustain that over a longer period of time. So I'm happy to have been able to share with you the Eyevensys approach to non-viral gene therapies for the treatment of ocular diseases. And I'm hoping that as we move forward, we'll be able to add to this list of potential treatments of both inherited retinal diseases as well as more common retinal diseases. And in closing, I'd like to share with you an analysis of our management team, which includes Patricia Ziliak, as well as our board of directors. Thank you very much.